All right, good morning, everyone. Welcome back to another week of Past to Present 2026. My name is Julia. I work in the education department here at Columbia River Maritime Museum. And this week, we are welcoming a couple of speakers from Columbia River Intertribal Fish Commission. And that was a beautiful reminder for me to tell you to please silence your cell phones before we get started. Appreciate that. So today we have Dr. Rosie Gradoville, who is Lead Oceanographer for the Columbia River Intertribal Fish Commission's CMOP program. She earned a BA in biology from Barnard College in 2010 and a PhD in Ocean Ecology and Biogeochemistry from Oregon State University in 2017, and now leads the CMOP Observatory, a long-term program based in Astoria that monitors the coastal ocean and the Columbia River estuary. Her team studies how physical, chemical, and ecological processes shape the estuary and nearby coastal waters, providing information that supports salmon restoration and other tribal priorities. We also have Charles Seaton with us this morning. He is the lead modeler for the Columbia River Intertribal Fish Commission's CMOP program. He has a BS in botany from the University of Massachusetts Amherst and an MS in environmental science and engineering from Oregon Health and Science University. His team focuses on modeling the physical processes of the Columbia River estuary, coastal ocean, and the full Pacific Ocean. He has worked on the Columbia River estuary at CMOP for 25 years across 3 different institutions. So with all that being said, please welcome Rosie and Charles.
Thank you so much. Hello, everyone. Good morning. I'm Rosie. This is Charles, and we are excited to be here to tell you about the work that we do in the estuary and ocean. So we work with the Coastal Margin Observation and Prediction Program, or CMOP, and our program has 2 sides to it. We have what we call an observatory, where we do long-term monitoring in the estuary and ocean using things like buoys. And then we have a prediction program where we take our long-term data at discrete stations and use numerical models to extrapolate what conditions might be like in the entire estuary and ocean in the past, present, and future. We have an amazing CMOP team, including 2 field staff members who are based in Astoria. So Dan and Andres, could you raise your hands? And we have a team of 7 working to study the system. We are part of the Columbia River Intertribal Fish Commission, which is another mouthful. We call it CRITFIC, and it's a tribal organization with the headquarters in Portland, working to restore salmon populations in the Columbia River. And finally, CMOP has a long history in Astoria, dating back to the 1990s. So these are the 4 topics that we want to tell you about today. And we'll start by telling you about our organization, the Columbia River Intertribal Fish Commission. So we work for the 4 Columbia River Treaty Tribes, and these are Waikonishpum, or salmon people. Salmon is one of the sacred first foods for our tribes, and they have a creation story that says that when people were first created, the salmon gave up his body to feed the native people. But in doing so, he lost his ability to speak. So there's now a reciprocal relationship where the salmon feed the people, and in turn, the native people must take care of the salmon and the habitat that supports them. And salmon have defined the diets, economies, and religions of tribal cultures in our region since time immemorial. Historically, salmon comprised half of the calories consumed by native peoples, and even today, tribal members consume fish at 10 times the national average. Abundant salmon and strong winds in the Columbia Gorge region allowed salmon to be an important trading commodity. And to this day, the return of the salmon each spring is a time of celebration, Thanksgiving, prayers, and feasts in longhouses throughout our region. Salmon are so fundamental and important to our tribes that when tribal leaders negotiated the treaties with the US government in the 1800s, they included the right to fish in those treaties. So the treaties include important language that says that they retain the right of taking fish at all usual and accustomed places. And that means that they can take fish on and off their reservation, and those are treaty protected rights. Here's a map showing the Columbia River Basin in the dark tan color and the ceded territories and current reservations of our 4 tribes, the Yakima, Warm Springs, Nez Perce, and Umatilla tribes in the 4 colors. So you can see that a lot of lands were ceded through these treaties. The ceded lands cover 25% of the Columbia Basin and 55% of the rivers and streams that are still accessible to salmon. So a lot of lands were ceded. However, those fishing rights were retained. And the fishing rights have been upheld in a series of court cases over the past 80 years or so. So for instance, treaty tribes are entitled to a fair share of fish, and that has been defined by the courts as 50% of the harvestable fish destined for those ceded territories. But sadly, salmon populations have drastically declined since those treaties were signed. We estimate that returning Columbia River salmon populations have declined by about 90%, from 15 to 20 million back in 1855 to just over 1 million in 2020. Some runs are facing the threat of extinction. There are 13 Columbia River runs currently listed under the Endangered Species Act. There are a few reasons for this decline. First, overharvest. The first commercial Columbia Basin salmon cannery opened in 1866, and by 1883, there were 39 canneries producing over 42 million pounds of salmon each year. There's also been drastic habitat decline for the salmon. There's been clear cutting, there was in-river log transport and mining that led to a lot of increased erosion and degraded stream habitats, negatively impacting salmon spawning, rearing, and migration. And then, of course, there's the series of dams in the Columbia and Snake Rivers. This is a picture of Celilo Falls, which was one of the most productive and culturally significant salmon fisheries in the Columbia Basin, but it was inundated by the Dalles Dam in 1957. And today, there is a huge hydropower system that has completely changed the way the river runs. From 1906 to 1975, 34 dams were built on the Columbia and Snake Rivers. This includes 3 major dams that don't allow for any fish passage at all, so it cuts salmon off from vast areas of the basin. But even dams that do allow for fish passage can cause bottlenecks that lead to increased salmon mortality. So, faced with the prospect of losing salmon, the 4 Columbia River Treaty tribes, the Yakima, Nez Perce, Warm Springs, and Umatilla tribes, came together in 1977 to form our organization, the Columbia River Intertribal Fish Commission, in order to coordinate their fisheries management and work towards reversing salmon decline. So here's that same map showing the basin and the ceded territories of our 4 tribes with the CRITFIC offices. So we have our headquarters in Portland, but we have other offices up and down the river. And we do work including policy, enforcement, there's CRITFIC police on the river, and then huge science and management departments. A lot of the science at CRITFIC happens in the freshwater systems. We have a genetics lab that does genetic stock identification, so they can take a fish and sequence its fish tissue and tell who its parents were. There's also work to improve hatchery practices, support habitat restoration efforts, and track things like invasives, toxics, and climate change in the rivers. But now, starting in 2020, CRITFIC acquired the CMOP program, a pre-existing ocean and estuary research program. So we now have our newest office in Astoria with CMOP. And the reason that CRITFIC acquired the CMOP program is because the tribes understood the importance of healthy estuary and ocean environments. This shows the salmon life cycle. Salmon, of course, are born in the freshwater, but they travel through the estuary twice during their life cycle, and they spend years out in the ocean. So we really need to understand and protect these systems to protect the salmon. And with that, I'll hand it over to Charles.
So I'll be talking about the history of CMOP, both before and since we've come to CritFic. So our program began in the 90s. We've really had sort of four epochs of the program. In the prehistory, our leadership was involved in previous research programs in the estuary, looking at the estuary turbidity maxima. And those programs were really focused on sort of temporary, short-term ship-based observations. So we'd go out for a week or two and do intensive observations in one or two locations, and then come back 6 months later and do another 1 or 2 weeks of observations. And you can piece out some things about the system from that, but it really leaves a great deal that you haven't explored. And so our leadership decided that what we really needed to do was to have multiple stations making long-term observations to be able to see what happens constantly in multiple locations through the system. And so in '96, we started collecting long-term continuous physical data, which we've collected since then. And then in the mid-aughts in 2006, we successfully got funding from the National Science Foundation to create the Science and Technology Center, which we call CMOP. And that was centered at OHSU, but also incorporated people at University of Washington and Oregon State. And then that NSF grant was only 10 years long, so when it ended in 2016, we reduced back down to a smaller program similar to what we'd had in the prior decade. And then in 2020, the leader of that program retired, and he worked to transfer our program to CritFic. In our STC CMOP days, we were very successful as a research institution, producing over 200 publications. And regionally, earlier even than when we became CMOP, we were a founding member of a regional association of ocean observing systems called NANUS. And they have really great resources. We'll show a little bit about that later at the very end. So we actually, the need for these long-term observations came in part from the desire to create models of what was happening in the estuary and the need to have data to constantly test those models against. If you only have little bits of data, then it's easier to build models that'll match that data, but won't work outside of that time period. So having constant, continuous data to test your models against forces you to really evaluate whether your models are working. Our program was originally led, and until he retired, by Dr. Antonio Baptista, who came out here in '93, participated in those early programs, and then decided that we needed the long-term observations. We were based originally at the Oregon Graduate Institute in Beaverton, which eventually became part of OHSU. For our models, we have lots and lots of computers to run them on. And we raised up generations of modelers, mostly spending their times in offices, but also occasionally being sent out on ships. Here is one of them, Sergei Frolov, practicing putting on a survival suit. It's good to send your modelers to sea so they know what they're actually dealing with. And we produced generations of models, gradually becoming better and better. And I'll talk much more about our models later on. So moving to the observatory side of our system, it really is impossible to talk about the development of our observatory without talking about Michael Wilkin, who sadly passed away last May. But he led our program for more than 25 years. And he was involved in all of the observatory side. He designed most of the observatory. He picked the stations. He worked with people locally to arrange things. He was just the core of building the observatory. He led our team of staff out here for multiple decades. He worked with researchers within CMOP and before. And he loved working with high school students and kids as well. Here he is with a summer camp that CMOP ran at one point. And he was a good friend as well. So some of those pictures I've shown already with him are from the training vessel Forerunner, which is run by the Clatsop Community College and based at the Mertz campus. We've had a multi-decade relationship with Clatsop Community College and with Forerunner in particular. And it's older paint colors of black and white, and it's more recent paint colors of yellow and white for anyone who knows the Forerunner. Mertz, it's a campus down on the water on the other side of Tongue Point. It stands for the Marine and Environmental Research and Training Facility. And the environmental research in the name really is based on its connection to Corey and then to CMOP. We were involved in the original grants that built the facility, and we worked closely. We were based there for many years. We launched our buoys off of the Forerunner. We launched our undersea robots called gliders off of the Forerunner. And we did extensive science from the Forerunner and within the hold of the Forerunner. So that was a really great relationship that we're working to continue. One of the things that just as a side note on things that we researched off of the Forerunner, the red water blooms that happen in the late summer and early fall. This is the Astoria Megler Bridge here, and this deep red in the water is the red water blooms that happen every few weeks in the late summer. It's this organism called Mesodynium major, and it swims up in the water. So when you pull it out of the water and stick it in a bottle, it'll swim up and form this tight layer. And that's what allows it to stay in the estuary and survive. It's just a really neat organism. So moving to the history of our stations, and Rosie will talk more about the actual what we get from our stations and how we manage them. I just want to run through some of the history of the stations. So looking at that photo, this is Cathlamet Bay. That's Tongue Point there. We're somewhere over in there. And all of these stars are places where we've had stations over the years. So we had a really extensive network. And then more recently, actually in the CMOP days, in the SDC CMOP days, we've narrowed it down to a smaller set of stations with more extensive instrumentation. So in the early days, we based almost all of our stations on the navigation markers around the estuary. They provide fantastic existing platforms. Michael ran a dive program. So we would put communications equipment up on the tops of the towers and then dive and install instruments down on the bases of the towers. We did this all throughout the estuary. We also established a buoy program, both initially offshore buoys and then more recently inside of the estuary. In the SDC and CMOP days, we also expanded upwards into Portland and established a station up there. And then we also, in order to be able to do the more complicated measurements that we wanted to do, it's very difficult to stick expensive instruments that have a lot of maintenance needs underwater and maintain them with a dive program. So we found places within the estuary, here a pier in Mott Basin on the east side of Tongue Point, where we've got permission to build facilities, sheds, and pumping systems to bring water up from the estuary to the shed, where we established instruments to be able to measure all of these variables with better maintenance. And then moving from this past to today, we're continuing to focus on the dock-based stations. This is Andres, our new field lead. And we moved in order to expand our facilities, we moved out of the Mertz campus and took up space in an industrial facility over in the east end of Astoria. building. This is before we started building out our new facilities. We're in the midst of building our new spaces. And this is our new field team for the past several years now of Dan and Andres.
Right, so next I'm going to tell you about our observatory. So this is a map of our, not photo map of our current observatory. I'll take you on a really quick tour. We have one ocean buoy. This is our largest and most complicated buoy. It's located in the Columbia River plume. Located in the Columbia River plume. We put it out in the spring and pick it up in the fall, and it collects measurements from 6 depths. Then we have had 2 pier-based stations. Here we pump water from multiple depths up to the shed at the end of a pier. These are great for long-term monitoring, easier to get to than going on a boat. However, I say that we have had to because sadly, after 15 years of monitoring, we had to vacate our Point Adams station when the fish packing plant closed down this fall. Then we have 4 buoys in the estuary in Baker Bay, Youngs Bay, Cathlamet Bay, and Elliott Point. These are smaller, simpler buoys where we just collect data from a single depth. And then we finally have 2 stations further upriver that we're working to reoccupy. At all of these stations, we make measurements. We have instruments that collect physical data like temperature, salinity, and turbidity, chemical data like the concentrations of dissolved oxygen, carbon dioxide, and nutrients, and biological data based on phytoplankton fluorescence. So we have buoys, we get our data, but there's really a lot of steps to go from buoys to data. So I want to take the next few minutes talking to you about how exactly this works, and then I'll step back and try to convince you that all of it matters and these data are worth collecting. So first, how do we turn buoys into data? It starts with a lot of preparation, and that preparation really begins with picking up a buoy from its past deployment. Here you can see some pictures of our plume buoy after it was collected. It's very dirty. It needs to be cleaned. It had a lot growing on it out in the ocean. The buoy also incurs a lot of damage being out there. So we start with cleaning, repairing, and doing forensics if something went wrong to try to tell what we can do better next time. And for this last plume buoy deployment, Andres and Dan made a lot of changes to the physical design and to the microcontrol board of the buoy. So I know this is too small to read all of the text, but hopefully you see there are many complicated components from the solar panels and radio up top down to the clump weight at the very bottom. So we have to code and wire the microcontrol board or the brain of the buoy. We have to test and install all of the scientific instruments that get, most of them go beneath the buoy right here. And then we have different lengths of tubing here going down all the way to the seabed. So we pump water from 6 different depths up to those instruments. We also need to test all of the pumps. And then we need to refurbish the hull, paint the buoy, and assemble all these components before it's ready to go out to sea. So once it's finally ready, we can deploy it. Here, the photo on the left shows the buoy on a trailer ready to go out to sea. And historically, we used the Forerunner vessel through the partnership with Clatsop Community College for this field work. But now that we've moved off of the Mertz campus, it's a little more complicated to coordinate. So we're starting to rely on our own CritFic vessels more for the small buoy deployments. Here you can see a couple pictures of our 22-foot Sea Dory and our rigid hull inflatable, the buoy on it here. But we don't have any of our own vessels large enough to deploy the ocean buoy. So for that, this summer, for the first time, we chartered with the Cape Windy. Here you can see the plume buoy going out to sea on Cape Windy. It's a local fishing vessel. And once the buoy is out there, we monitor the data to see how it's doing. This is a vastly oversimplified diagram showing the flow of our data from individual instruments all the way to our data repositories. And there's a fun step in here where the data gets sent by a radio network of 900 megahertz radios. So this is what our radio network looks like in the estuary. You can see a picture here of our little antenna up here. And that radio network sends the data back to shore to our field office based near Pier 39. And there the data go to local computers and they're up on servers and processed in a matter of minutes. So we can then monitor the real-time data to see how the buoy is performing. And when we start to see big data gaps or we see evidence that an instrument isn't performing well, then we try to find a time to go recover the buoy. So for recoveries, here are a couple pictures of our plume buoy recoveries. This is the most recent recovery using Cape Windy this past fall. And here's a previous recovery on the Forerunner. But I want to tell you a little story about when a ship wasn't available when we needed it to recover our plume buoy. And that was back in fall of 2024. You might remember we had some very bad weather that fall. One of the types of data we monitor is our GPS data to know that the buoy is where we expect it, where we put it. So that's what this graph is showing you here. The y-axis is offset from station in meters, and the x-axis is time for November 2024. And we experienced some huge king tides followed by this bomb cyclone, and that ended up walking our buoy about 3.5 kilometers northwest away from its original location. And it actually moved close to the edge of the navigation channel and became a safety hazard. So we were in contact with the bar pilots and the Coast Guard. We tried to find a boat to charter us immediately, but one was not immediately available for recovery. So we were very fortunate that the Coast Guard came to the rescue and picked up our buoy for us. And it came home safe and sound by mid-December 2024. All right, and then finally, we curate our data. We have our data available as real time after some initial processing, and we also put all of our data under vigorous quality control. And it's then available on our website and also on the website of NANUS, the Northwest Association of Ocean Observing Systems that we're a part of. So if you're interested in looking at current ocean conditions, you could check out either our website or this NANUS website that also hosts a lot of other regional data. Okay, so I think I hopefully demonstrated that it's a lot of work to make these measurements. So why do it? Why does it all matter? Well, these physical and chemical and biological conditions of the water, or what we call the biogeochemistry of the water, are the type of data that oceanographers collect all across the world to help detect change in the oceans, improve forecasts, and support decision-making. So we're part of a global community trying to learn about our oceans. But on a more local and personal scale, these are all water quality data that directly affect salmon survival in the estuary and the ocean. So for instance, salmon need cold enough temperatures to survive, and salmon need enough oxygen to breathe. So I'm going to take a few minutes and tell you a little more about why we study dissolved oxygen, or why we track dissolved oxygen and temperature, and how that our data indicate that they might be affecting salmon. The reason we need to worry about dissolved oxygen concentrations along our coast and in the estuary is because of this process called coastal upwelling. When in the summer along our coast, winds tend to blow from the north, and those winds combined with the rotation of the earth pull surface ocean waters away from shore and bring deep ocean waters up to shore. So we have the opposite process happening in the winter. We tend to have winds from the south, and that causes downwelling, causing surface waters to come to shore. So in the summer, when we have upwelling happening, that brings waters that have really high nutrient concentrations. So in a way, it's a good thing because that fuels primary production by phytoplankton and supports our productive fisheries. But those deep ocean waters also have really low dissolved oxygen concentrations. You can't have photosynthesis in the deep ocean. So we sometimes see really low dissolved oxygen concentrations, or what we call hypoxia, off of our coast that can lead to mass die-offs of marine organisms. And we expect that this process will worsen with climate change. We're seeing evidence for more and more severe upwelling-driven hypoxia off of our coast. So this is one of the processes that we monitor at CMOP. We collect dissolved oxygen data in the ocean and in the estuary. So I want to show you a little bit of our data. Here we're looking at dissolved oxygen concentrations at our plume buoy, our ocean buoy. So again, here we're looking at dissolved oxygen in the color with high oxygen levels in yellow, low in blue. And we're looking at a profile, so where depth is on the y-axis and time is on the x-axis. So we're just looking at what the oxygen levels were doing over summer 2022 at our plume buoy. And you can see that in these deeper waters, dissolved oxygen concentrations started to get really low later in the summer. And that's driven by the wind patterns. Here we're looking at wind speed with the blue colors showing north winds and the red showing south winds. So as upwelling favorable winds intensified later in the summer, we started to see really low dissolved oxygen in our coastal water. And the tides bring that water into the estuary. So we also see upwelling driven hypoxia in the estuary. Next, I'm going to show you some dissolved oxygen data from our Point Adams station, which is near the river mouth, we're near the river mouth. And that's what we're looking at here. Now we're looking at many years of data collected at Point Adams, and dissolved oxygen concentration is on the y-axis and time on the x-axis. I know that this is a lot to look at, but I just want to point out two things. We're looking at 15 years of data, and you can see this strong oscillation with higher oxygen in the winter, lower oxygen in the summer. And we also see that when the dissolved oxygen is low, the salinity is high. So in our estuary, when we see hypoxia, it's driven by the intrusion of upwelled coastal waters into the estuary. All right, so we have so many years of data from this station that we can make what we call a climatology, or look at seasonal patterns and understand what's normal. So now I'm showing you the same data, dissolved oxygen at Point Adams. Again, we're looking at dissolved oxygen on the y-axis, but I've plotted the data according to day of year, and we're looking at the average range from all 15 years of data here in the dark gray. So when we look at the data this way, we can see there's clearly a consistent pattern where dissolved oxygen concentrations drop each summer, driven by that coastal upwelling, and they always drop low enough that they could cause potential physiological stress to salmon. That's what the color coding is representing back here. So we can use this understanding of what's normal to make sense of what we see in a current year. So now the blue color is showing you our observations from 2025. So we can see that in 2025, we had a long time from late May all the way through to late September, where dissolved oxygen concentrations at Point Adams were quite low enough to cause stress to salmon. So we had an elongated period of hypoxia this year. Okay, now I want to show you the same type of plot, but for temperature data, because we know that temperature also matters for salmon. They need cold water to survive. So now I'm showing you the same type of seasonality plot, but this time from our Tongue Point station. We're looking at surface water from Tongue Point. That's pretty reflective of main stem Columbia River temperatures. And we see, of course, temperatures warmer in the summer, but we see that at this station, temperatures always rise warm enough to cause thermal stress to salmon. And when we look at our 2025 data, we saw that we had very warm temperatures in June and again in August and September. We actually set a record for the warmest on days in August for the warmest temperatures we had ever recorded at this station. So it's an especially warm year. Now, finally, I'm just going to show you these two plots we already looked at. And, so again, we had hypoxic conditions from June through September last year, and abnormally warm water in the spring and the summer. One of the main reasons for the warm water last year is because it was a low flow year for the Columbia River. The river flow is highly regulated, and it changes a lot from year to year, as Charles will talk about. This graph shows the average river flow for this past 15 years in gray. And then it shows you that 2025 was a year where there was not much cold water flowing down the river. So it's part of why it was so warm in our Tongue Point station. And again, the wind patterns and the process of coastal upwelling is really what drives the hypoxia. So this was a year where we saw really consistent north winds in the summer, and that led to this long period of hypoxia that we saw in our stations. So that's all I have to show you for the observatory. This is really just an example to show you why we make these long-term measurements so that we can understand the system and make sense of what we see today.
So finish up by talking about what we were able to learn, what we do in our modeling system, and what we're able to learn from that. So our modeling system is, we call it the virtual Columbia River. We run it both as forecasts, similar to weather forecasts, except for what's happening in the water in the estuary. We also run it for long periods of time, running back into 20 years in the past, and then we construct those same sorts of climatologies that Rosie was just describing, except based on what we see in the model rather than observatory. And it also has been used to support search and recovery after maritime accidents. So what do I mean when I say that we have a model? And sometimes I'll call it a hydrodynamic model to distinguish it from other kinds of models. So what we're doing is we're describing what happens from the river into the ocean, solving the physics equations that describe the motion of water. So water piles up if more water is coming into a place, raising elevations. Water runs downhill. Denser water will flow underneath less dense water, and stuff in water will move with the water. Those are essentially the basics of the physics of how water moves. But the equations are quite complicated, and in order to solve them, we have to simplify the world and simplify the equations. And we do that by describing the world as a mesh of triangles and quadrangles. And that allows us to have a simpler world, but one that still can efficiently follow the shape of complicated channels within the estuaries. And because the density of salt water and river water are different, so salt water will flow underneath river water, we have to look at the three-dimensional structure. So we extend these triangles and quadrangles down into stacks of vertical levels and have a world that's constructed of prisms of different shapes. And then we solve physics equations on those, and we do it for a minute. We determine what happens a minute later, and a minute later, and a minute later. And we run that to cover days in the case of forecasts, or decades in the case of the hindcasts. So what are the models good for? There's a famous, at least amongst modelers, saying that all models are wrong, but some models are useful. So we have to look at what does the model get is good enough at that we can trust it to say something meaningful. And then we have to look at how can it be used based on what it's capable of seeing. So one of the questions that we look at in the estuary, both from a perspective of management and a perspective from understanding our models, is how far up the estuary does salt water reach? The areas that are freshwater, particularly freshwater wetlands, are very different from and much more productive than the wetlands and brackish areas. So it matters a lot how far salinity is reaching. So we can tell with observations at particular locations how far does salt reach. And in this diagram, the ocean water is this red line. The vertical here is how salty the water is, and then the horizontal is the calendar month. And the different colors are different years. So this is fairly far upriver within the lower portion of the estuary, up towards the the eastern end of Kathlamic Bay, right in the main channel. And we can see that in the late summer, running into the fall and even a bit into the winter, we frequently see quite high salinity, more than a 50-50 mix of ocean water and fresh water. And then earlier in the year, there's a period where we basically never see salinity, and then there's the earlier period where we occasionally see salinity, but mostly we don't. So that's what the observations can tell us about a specific place. But the models, the virtue of the models is that they describe the entire estuary, so they can tell us where the salinity is when it's not reaching this particular station, and they can start to tell us why the system is behaving the way that it is. So one of the fundamental things that we have to represent in the model is what makes the estuary what it is. And the basis of the estuary is its shape. So what are its channels shaped like? How deep are they? How are they connected? How large are the tidal flats? what structures at the mouth are narrowing the channel. All of this information needs to be represented in the model as the basis of building the model. And then we have to look at what drives the estuary and then implement that into the model. So the two big things that drive the estuary is where the river meets the ocean. So the two things that are driving it are the flow of the river. So this is the top figure here is the flow of the Columbia River at the Bonneville Dam. And we can see the spring freshet and the low flows in the late summer into fall. And then the second thing that's driving the estuary is the ocean in the form of the tides. So this is the tides is measured at Astoria, at Tongue Point. And we can see the fluctuations between neap tides and spring tides. So the large tides and then the small tides. And then if we look at what's happening in this particular year, this was 2020, at that Elliott Point station, again with the salinity, we can see that there are these pulses of high salinity coming in. And we get one big pulse a month and one little pulse a month. And so we have this period where there's no salt reaching, and this is basically the freshet. And then we have the period where salt does get to Elliott, and that's the period of low flow. And then dividing line is roughly around there. And then we have these sort of outer winter, spring periods where we don't really see much salinity, even when the flow at Bonneville is relatively low. And that's because there's actually a lot of flow that if you're looking at Bonneville Dam, you're missing all of the rain-driven flow coming in from the smaller rivers downstream. So the flows really mostly here are not good enough, are not low enough to allow salt water to come up. And then if we look at it compared to when these spikes happen compared to the tides, we'll see that they happen during the neap tides. Particularly the most extreme neaps give us these large spikes, and then the little spikes happen when there's sort of not as strong not as extreme of neap tides. So looking at what we can see in the model, this is the sort of visualization of the detailed structure of the world that we can see in a model. So this is kind of a noisy figure. It's taken from the daily forecast. This is yesterday's forecast, so covering yesterday, today, and tomorrow. And I'll go through this a little bit. So the top left figure is just showing the tides influencing the model, and that red line is showing where all of these other figures are within the tide cycle. This lower left figure is a map of the estuary. We're about there. And red is the ocean water, and dark blue is fresh water, and then the sort of intermediate rainbow is intermediate salinity. And there are these three black lines running through this map, and those match with these three other figures on the right. So each of these figures on the right is a transect. And then in here, we're looking at the vertical structure of salinity along that line. So the top here is the surface of the water, and the bottom here is the bottom of the river. And we can see coming from the riverside, this fresh water coming on the surface, and from the ocean water, salty ocean water coming in at the bottom in these pulses. And then we can see the difference across these three different transects in this figure. But each tide, we get a pulse of salt water coming in, and then on the flood tide, and then a pulse of fresh water pushing the ocean water back out on the ebb tide. So Elliott Point is not within this map here, but if we look at another figure, this is expanding upward across the entire estuary. So Elliott Point is here. We are here. This is today's forecast. This was the high tide at 6 o'clock this morning. So you can see, according to our model, the water at the bottom of the river just out there was about a little less than half salt water. So if we move to looking at 4 different times of the year back in 2020 when we had Elliott Point data available, we get these 4 different conditions under spring tides, neap tides, high flow, and low flow. So the low flow and neap tide is the period where we see in the observations that we're getting salt all the way to Elliott. And within the model, we also see that this is the period where we get the greatest extent of salinity up the main channel. So we get much less salinity, much less salinity, and much less salinity under both the high flow conditions and the spring tide conditions than we do in the neap tide low flow conditions within the model. So that's consistent with what we're seeing in the observations and tells us that we have some useful information in the model about salinity intrusion within the estuary. One thing you may notice, though, is that Elliott Point is here, and the water right there, even in these low flow, low tide conditions, is not actually reaching to Elliott. So the model is wrong, but it's still useful. Additionally, these are the forecasts, and the forecasts are really about 16 years old, or less, a little less than that, 10 or 12 years old since we developed the forecast model, and we've continued to work on and improve the model since then. So I'll just show one example of comparing our newer, better model, which will be implemented as the forecast sometime quite soon. So here again, this is at a low flow, weak tide, neap tide period. The forecast model that I was just showing, similar behavior. It makes it reasonably far up the channel, but it's not reaching Elliott. In our latest version of the model, we're able to push salt substantially further upriver and actually to reach Elliott. So the model is less wrong and probably more useful. So one of the great things that we can do with the model is that we can ask what if questions. So because the model is just an imagined thing within a computer, we can change the depths within the river, and we can see, does salt get further up the river? Does salt get less far up the river? We can change river flows. And these reflect things that have happened in the real world or that will happen in the real world. So back in the early aughts around 2000 and a few years after that, there was a huge argument over whether to deepen the navigation channel in the Columbia River to allow larger ships to make it upriver to Portland. And there was a great deal of concern over whether, if we made the channel deeper, would more salt water come in and would it start to infiltrate into Cathlamet Bay and kill off the freshwater tidal wetlands in Cathlamet Bay. So there was a huge fight between different agencies within the government and different organizations within society. And we eventually settled the issue with our models, looking at what happens if within the model we deepen the channel. And what we found was that it didn't increase salinity intrusion. And that was the basis on which the region decided to deepen the navigation channel. And then we used our observation network for a decade after that to monitor what happened in the estuary to make sure that that was actually reflected by reality. And indeed it was. We didn't see any changes as the channel was deepened at our stations in Cathlamet Bay or at Elliott. And then when we reran the model, when it was a better model then, to confirm that the model said the same thing in a more advanced form, and it did. So that's what we're able to do with the models. We can also apply it to looking at how things will change if we change how the river is regulated or how it will change under climate change and sea level rise. So the model has been used to look at salmon, understanding how salmon move through the estuary and how they use the estuary to look at habitat identification, particularly defining where those freshwater wetlands are. It's been used in coastal resilience, both actually in the tsunami studies in the aughts, and then also used in the FEMA evaluation of the dikes in Warrenton to see whether they'll be able to survive a 100-year storm and flood event. They've been used for search and recovery efforts, and they support navigation. Some new things that we're doing with the model. One really neat thing that we've been working on for the last 5 years is a model that expands includes the full detail of the virtual Columbia River, but incorporates it into a model that covers the entire Pacific Ocean. And so this was developed in collaboration with NOAA, and it's becoming an operational forecast this spring. Its name is the terrible mouthful of STOF's 3D Pacific. So we are also working, we have a project currently that we're working on doing new work on combined flooding and coastal resilience, particularly looking at Baker Bay and Grays Bay and the Grays River. And for that, we're incorporating much more detail of local sources of fresh water into the estuary, and then looking at what-if scenarios of how can we incorporate nature-based solutions to flooding to decrease the combined flooding problem while not threatening salmon habitat. One last thing that we're doing is we're working with a research group at Oregon State led by Mick Haller to develop wave models that use the CMOP virtual Columbia River's currents. And so this is a need that's been expressed by the Bar Pilots Association, particularly for a very long time, and is finally actually being developed. So the figure on the left is currents with yellower color, green to yellow being stronger currents. And then the figure on the right is waves with the orange to yellow being stronger waves. And so you can see that the patterns within these two figures are very similar. As we get a jet of outgoing water in the estuary, forming these strong currents flowing out, the waves pile up as they fight against these currents. And that's one of the things that makes the Columbia River Bar such a dangerous place. And so by combining the modeling of waves with the modeling of currents, we hope to be able to provide better support to the bar pilots and to other boaters crossing the bar. So concluding with that, we study the ocean and estuary with data and models. If you'd like to learn more, there are both the CMOP.critfic.org website and the nanus.org website, which also gives you lots of other people within the region doing similar work. And you can learn more about CritFic at critfic.org, the main website of our organization. And additionally, there's an email mailing list that sends out relevant news about the Columbia River and the tribes every day. That is a really great resource, and you can find a link to that on the critfic.org website. And we have a field office here in Astoria, and come say hi and learn more about us. Thank you. And I acknowledge our former staff and collaborators and funding sources. Thanks.
Thank you so much. It's fun to see how the subtitles are figuring out CritFic. I really appreciate you both taking the time. So Katie and I both have handheld microphones, and we'll walk around the room for about 5-10 minutes with questions folks have. If you just raise your hand and we'll come around and grab the microphone for you. I have a question over here in the corner.
My question's on the red tide you're talking about. I work out of the night shift. I get asked that question. I know it's only for about a month. But what I get asked is it pollution? Is it toxic? And is it food for fish? Can you give me something to tell them?
Sure. It is not toxic. It is not pollution. They are, is there food for fish? I don't know if they're food for fish. Something must eat them. There's a lot of them.
Plankton.
Yeah, they're plankton. They actually swim. They're really neat organisms. If you look them up and find videos of them online, they swim by hopping. They're just really cute creatures, and they I don't know if we do.
The
We
old
should.
website
The old
does.
website does. I should. Yeah, I should track it down. And they're super neat. They don't do photosynthesis themselves. They eat another kind of phytoplankton. And they keep the part of the phytoplankton that does photosynthesis and do photosynthesis that way. But it's called perieclepsy. They steal the chloroplasts.
All right, we have a question in the back.
Yeah, we know that bottom waters, like you said, are oxygen depleted or have low oxygen. However, they're nutrient rich. Also, the bottom waters are nutrient rich. So I don't understand the interplay of the low oxygen with the high nutrient contents that's coming from the bottom water. We know our coastal waters are great for fishing. So it seems like the nutrient rich aspect dominates the oxygen poor aspect.
Right, yeah, it's a great question. Yes, so it's a two-sided coin, the fact that we have coastal upwelling. If we didn't have that, like you said, we wouldn't have such high nutrient concentrations, we wouldn't have such productive waters. And so the fish ultimately need that process of upwelling to bring the nutrient rich water to the surface. But at the same time, it can bring waters that have such low oxygen that they can harm some species, especially like crabs or other bottom dwelling species that experience, that are near the coast and experience this pulse of really low oxygen water coming up. Those deep waters also have high carbon dioxide concentrations. So in terms of ocean acidification, worrying about the low pH corrosive waters, that those upwelling events can also cause corrosive conditions for the shellfish. So it really both helps and hurts the organisms. And one other detail we didn't talk about is that during coastal upwelling events, there'll be the upwelling pulse that brings the nutrients to the surface. That'll make phytoplankton production, all this productivity at the surface. Then there'll be a relaxation of the winds. Those phytoplankton will sink and decompose, and that will draw oxygen concentrations even lower. So a lot of oceanographers study those events in detail, the interplay between productivity and hypoxia.
Are you monitoring the pollution coming out of Hanford?
We aren't. I don't know who is. We actually did participate. There was a study not looking at Hanford, but looking at pollution more broadly that we participated in. The Estuary Partnership was the lead on that, but we deployed detectors at the Point Adams station as well. So there were 10 stations in the last 2 or 3 years where we were monitoring mostly for organic pollutants rather than for radioactive. There is nitrate monitoring in the river. It's not as much of an issue out here. We just don't have as much heavily fertilized agriculture as, say, the Mississippi does. At least in the estuary, our main source of nutrients is actually the upwelled water rather than the river.
Maybe a question over here.
Not really a question, it's just, if I went to dinner with you all, I would not have anything I could say because I wouldn't understand a pretty word. So, only question is, I get asked this, and I always say the depth of the channel itself is 54 feet at the beginning, and it narrows down to 45 feet along the way. Is that true?
The minimum depth of the river, well, the minimum depth of the navigation channel, yes, that's up to Vancouver.
Right.
There are deeper spots, much deeper spots, like the Tongue Point hole, right off of Tongue Point. It gets much deeper than that, and there are lots of other spots upriver where it is deeper than that. But they dredge constantly to keep it to that 43 feet.
They monitor it and
Yes, sorry, yes, that's.
So it's 54 feet at the beginning,
Mhm.
and it's just kept. And they probably know different areas in the river which are going to be a problem. That's probably where they go first.
Yeah.
It's not going to be anything wrong.
I have a couple of questions.
Okay.
So red tides on the east coast, like in Florida, are any different than red tides on the west coast?
Yes. You wanna?
So red tide is often a general term that's used to describe all kinds of harmful algal blooms. Sometimes they actually are red. They're oftentimes not associated with the tide, so it's a funny term, but we all use it, right? So yeah, in this case, we have a totally non-toxic red water event that happens when this mesodynium blooms. And that's actually, if anything, maybe a positive thing for the river because it's photosynthesizing, making lots of oxygen. But yeah, on the East Coast, they have harmful algal bloom red tides. And then on our coast, we have harmful algal blooms that aren't usually red. So we do have some, toxic plankton that bloom off of our coast that can cause problems, but tend to not be red. So it's totally different species that in different areas that can cause those harmful algal blooms.
And this gentleman asked about Hanford, and I have a question about Trojan. Have you guys done any kind of work with that?
No.
And there's a limit on the number of fish people are supposed to eat. And I didn't hear any kind of issue with that. I mean, is that something anybody said?
Yeah, there's a lot of work going on toxics monitoring, and we'd like to be doing a lot more than there's currently funding for. But yes, that's more of an issue with the fish that stay in the river than it is with, say, salmon. Not saying it's not an issue with salmon, but it's more of an issue with the
Well, I think it's more of an issue with sturgeon.
yeah, it's an issue with sturgeon, and it's an issue with lamprey as well, because they spend a fair amount of time in the river and they have very high fat content, which helps to collect some contaminants.
I think this is gonna be our last question.
I was wondering about the effect of invasive plant species in the estuary, the slowing the water flow, and how that might come into play.
I don't really know that much about invasive plant species in the estuary. It's a problem that we've been looking at a lot. The CRITFIC has been looking at a lot further upriver. We've had a huge expansion of aquatic vegetation that was never there before. But I'm not as familiar with it, and I'm not really familiar at all with it in the estuary.
I noticed it.
Okay. Uh-huh. Okay. So is that sort of like the Eurasian milfoil and like parrot feather? Okay, yeah, I think that's okay, yeah. Yeah, that's the same thing we've been looking at further upriver as well.
That's being studied then.
If anybody's studying it down here, I don't know who is. We aren't directly.
Before we thank Charles and Rosie for their time, I do want to make you aware of our lovely save the date half sheets for the rest of the past present lecture series. This is week 2 of 5. Next week, we are going to have Captain Thomas Thomeyer. His family actually founded Tidewater Barge Lines, and he was a Columbia River pilot for many years. So his presentation is called Born to be a Slow Pig. And that is next Tuesday, February 3rd, same time, same place. I'm sure Charles and Rosie can stick around for a few minutes if you have additional questions, but otherwise, thank you so much for coming and thank you for your time.